Okay, thanks. I'm excited to wrap up this great session. So I'm going to talk to you guys today about some work that we've been doing at high resolution to look at layer-dependent activity in human prefrontal cortex during working memory. So to start with the theoretical motivation for this study, we know that working memory usually involves a series of functions. So we need to encode a sensory stimulus, we need to maintain a representation of that stimulus over the course of a delay period, and then typically we will use that to guide behavior in some way. And we also know from a wealth of literature that working memory has been linked to dorsolateral prefrontal cortex, and this is true in both humans and non-human primates. And there's some early evidence, especially from the electrophysiological work in monkeys, suggesting that these subfunctions may be preferentially localized to distinct cortical layers. So specifically, that delay-related activity takes place predominantly in superficial layers and specifically in the pyramidal networks in layer 3. While the response-related activity takes place predominantly in deeper layers, namely layer 5. But to date, there's no direct evidence for this dissociation in humans. We also had a methodological motivation for this study, which is that recent advances in fMRI have combined to allow us to achieve layer-specific functional resolution. I have layer in quotes here because we typically still can't resolve all 6 canonical cortical lamina, but we can start to divide things into more superficial and deeper layers. And to date, these methods have been applied in an exciting array of studies that have mostly focused on primary sensory cortices, so V1, A1, M1, and S1, lots of beautiful work there, but there's been little to no work in higher order association regions like prefrontal cortex. So our main question and our main goal for this study was to see if we could develop and apply layer fMRI tools to show layer-specific activity in human DLPFC during different periods of a working memory task. So I'll walk you through our task paradigm and hypotheses. We used a pretty well-validated working memory task. We had 2 different trial types. The first one was aimed at dissociating manipulation from maintenance, specifically during the delay phase. So the way this worked was participants saw a string of letters, and then they saw a cue telling them either to alphabetize those letters, so to rearrange them into alphabetical order, or to simply remember them, maintain them in the order that they saw them. So those would be the manipulate or maintain conditions respectively. And they did this over the course of a delay period, and then a probe letter came on the screen, so they had to make a button press response to indicate the serial order that that letter would appear in either the alphabetized or the original sequence. And that's the response phase. So again, this trial type is aimed at dissociating manipulation from maintenance, specifically during the delay. We introduced a second trial type, calling action versus non-action. So this looked very similar to the first trial type, except now when participants get to the response phase, they see one of 2 different probes. So they either see a real probe that requires an actual button press, or they see this dummy probe where they don't have to actually do anything. But importantly, they don't know which of those trial types it is until they get to that response phase. And it's also important to note that all of these trials are alphabetized trials. So this trial type was aimed specifically at dissociating action from non-action at the time of the response. And using these 2 contrasts, we had hypotheses about the interaction between layer, trial type, and trial period. So I'll walk you through those. So what we thought we would see, again, based on the monkey electrophysiological work, is that in the upper layers, these layers would come online and their activity would remain high, specifically during the delay period in those alphabetized or manipulation trials, whereas we should see less activity during the delay phase in remember trials. And this difference here, we hypothesize, is coming from those recurrently connected networks of layer 3 pyramidal cells. In contrast, the deeper layers should be potentially less sensitive to the delay period, but we should see a peak of activity in the deeper layers at the time of the response, but specifically in the action trials and not in the non-action trials. And this may be reflecting local networks in the deeper layers, as well as input from and output to premotor areas. So I'll tell you a bit about our acquisition setup. We did all scanning at 7T. And we're using simultaneous or near simultaneous contrast here. So the first contrast is the familiar bold signal, so blood oxygen level dependent. This is a sensitive contrast, but it's less spatially specific. So it's prone to draining vein artifacts, specifically at the surface. Our second contrast was vaso, or vascular space occupancy. So this is a cerebral blood volume sensitive contrast, which is less sensitive than bold, but it's more spatially specific. And we acquired these two contrasts in an interleaved fashion over the course of the run, and then we later sort the frames and analyze them separately. And most of the data I'll show you today come from the vasocontrast, since that is the more spatially specific one. A few other unique features of our scan setup. So because prefrontal cortex is so large and heterogeneous across subjects, it's difficult to pinpoint anatomical landmarks in everybody. So we actually ran an online functional localizer where the first 6 minutes of every scan were a standard resolution functional run where we were, and then we zoomed in on sort of the hotspot in DLPFC for subsequent slice positioning for the high resolution experimental runs. We used two high-resolution protocols over the course of the study. The first was an axial-style readout. You can see the specs on the screen there. We primarily used this for quantifying and comparing activity across layers. Later in the study, we introduced an even higher resolution protocol. It was a sagittally positioned slab, and we primarily used this to visualize layer-dependent activity in individual subjects. The way the task worked was we had both trial types in a pseudo-random order, 20 trials in each run. We acquired 3 runs of the alphabetized remember contrast and 2 runs of the action non-action contrast. And then we do all of our analyses in individual participant space. So we draw layer masks for each subject within our ROI that comes from the functional localizer. So we had one superficial, you can see in green in this example subject, and one red. And then we average the time courses in those layer masks for each trial type in each condition. So for every subject, we get a mean time course per layer per condition. Okay, so here are our results. I'm going to put the hypothesis there on the left so that we can refer to it. So when we look here in the superficial layers in this top row, if you look at the left-hand panel, the blue time course is alphabetizing, the green time course is remembering. And so you can see that nicely in line with our hypothesis, you see this difference specifically during the delay period where the superficial layers are more active, but only when the trial calls for alphabetizing or actually manipulating the information in working memory. And there's essentially no difference in the superficial layers between action and non-action. Again, recall that both of these trial types were alphabetized trials. So it seems that the superficial layers are sensitive specifically to manipulating during the delay phase. If we take a look at the deeper layers, we see sort of the opposite pattern of response, so we don't see much difference at all in the deeper layers between alphabetized and remember trials, but we do see this strong peak of activity at the response time period, specifically in action trials, and it's not present in non-action trials. So again, suggesting that the deeper layers are sensitive to the response phase, and specifically when it calls for an action. We can quantify this, again, with two-way ANOVAs, and so the statistics here confirm exactly what we saw from the time courses. So we do see an interaction in the superficial layers where they're more active during the delay, but only in alphabetized trials, whereas the deeper layers are more active during the response, but only during trials that call for an action. We can quantify this in a related but slightly different way, so we can actually subtract the time courses of one trial type versus another. So in these graphs, again, the top one is the superficial layers, the bottom one is the deeper layers. The purple line here is the difference between alphabetizing and remembering. So you can see that that difference peaks in the superficial layers during the delay, whereas there's not much difference at all between action and non-action. That's the teal line in those superficial layers. Whereas down in the bottom graph, you can see a big peak in the teal line, so a large difference between action and non-action, specifically at the time of the response. And again, if we quantify sort of the differential levels of activity and directly compare across layers, we can see that this interaction holds statistically as well. So the last piece of data that I'll show you are the single subject activation maps. So these come from, again, our even higher resolution sagittal protocol. So this is a map from a single subject. On the left-hand side is the bold contrast, in the middle is the vaso, and the cortex is sort of, you can see this convolution. I'm not even going to try to mess with this pointer, but hopefully you can tell sort of the axis of the cortex, and so the voxels in green, that's the contrast between alphabetizing and remembering. So you can see that that's localized to the superficial layers, whereas the red stripe underneath it is when we compare the signal during responding to baseline. So there's this nice striping pattern that's visible both in the bold and the vaso data. On the right is the layer anatomical mass that we use for this subject. So again, that's just one participant. We ran 5 people through the sagittal protocol, and you can hopefully see that even though there's some anatomical differences across subjects, we see this nice striping pattern where the green is on top of the red in pretty much everybody that we look at. So to summarize, the way, again, that we're interpreting this dissociation here, so we believe that superficial layer activity during the delay period may reflect that local recurrent excitation among the networks of layer 3 pyramidal cells. It may also reflect some long-range cortico-cortical connectivity, specifically with other areas involved in working memory like parietal cortex. The increased activity in deeper layers during the response may be related to motor control, so action initiation and monitoring could also reflect some feedback mechanisms. And I'd like to emphasize the technical achievement here. So we're very excited to have achieved layer-specific fMRI in a higher order brain area, and I think this will open the door to examine more directed connectivity inputs and outputs, and more specific cortical, meso, and microcircuits using high-resolution fMRI, so non-invasively in humans. So we're very excited to carry this work forward. In the future, we're hoping to look at additional tasks as well as functional connectivity between layers, and also take this into populations where these circuits are hypothesized to be disrupted, so specifically schizophrenia and other mental illnesses where we see working memory deficits. There's a preprint out on this. I have a poster later this afternoon. I'd like to acknowledge these are our collaborators and also my co-authors who are on the first slide, and specifically Renzo Huber, who's at Layer fMRI on Twitter. If you don't know him, he's a physicist that was responsible for a lot of the technical expertise here, and thank you for your attention.